Good morning, students. Welcome to B.Sc-II 4th semester Zoology class. So today I am going to discuss on another important cell organelle present in eukaryotes or animal cells, lysosomes. So lysosomes are spherical membrane-bound organelles that are generated by the Golgi apparatus. They contain hydrolytic enzymes and so function as part of the recycling system of the cell. So lysosomes are spherical structures and they are membrane-bound organelles that are generated by the Golgi apparatus. So by the Golgi apparatus, the lysosomes are synthesized and they contain hydrolytic enzymes. Lysosomes contain hydrolytic enzymes. Therefore, they involve in digestion of food substances present in the cytoplasm. So any foreign bodies that are coming into the cell, they are killed, digested by the lysosomal enzymes, hydrolytic enzymes present in the lysosomes. And structure. Lysosomes are acidic membrane-bound organelles found within cells, usually around 1 micrometer in length. Lysosomes contain numerous hydrolytic enzymes which catalyze hydrolysis reactions. So it has several hydrolytic enzymes which are required for the hydrolysis of several food materials, nutrients. The membrane surrounding the lysosome is vital to ensure these enzymes do not leak out into the cytoplasm and damage the cell from within. So the lysosome is also referred as suicidal bag. If the membrane of the lysosome is broken, the content of the lysosome is released into the cytoplasm. It may kill other cell organelles. Therefore, the lysosomes are called suicidal bags. In order to maintain the acidic pH of the lysosome, protons are actively transported into the organelle across the lysosomal membrane. To keep the pH low in the lysosome, acidic pH, the protons are actively being transported into the organelle across the membrane. See here, so this is lysosomal membrane. So ion channels are there. Protons are continuously being pumped into the lysosomes so that the internal environment of the lysosome is acidic. pH is lower than 7. Right? So this has acidic environment inside. To maintain acidic environment inside, the protons are being pumped across the membrane continuously. So in order to maintain the acidic pH of the lysosome, protons are actively transported into the organelle across the lysosomal membrane. Next, synthesis. The lysosome and the enzymes within it are synthesized separately. The lysosome and the enzymes within it are synthesized separately. Lysosome is synthesized separately and its enzymes are also synthesized separately. Lysosomal proteins are formed in the same way as any other protein. As any other protein being synthesized, in the same way, the lysosomal proteins are also synthesized. For synthesis of protein, what is required? Ribosomes are required. Ribosomes are attached to endoplasmic reticulum. So rough endoplasmic reticulum synthesizes protein. Then later on it is sent to Golgi complex. From Golgi complex, lysosomes are formed. The first step is the initiation of mRNA strand production from relevant DNA segments. Yes, for the synthesis of protein, message has to come from the DNA through mRNA. mRNA carries message for the synthesis of particular protein. to the cytoplasm. Clear? The mRNA strands proceeds to the rough endoplasmic reticulum where ribosomes construct the hydrolytic enzymes. You know, all enzymes are proteins. Okay, so hydrolytic enzymes are synthesized by the ribosomes. Later on, they are, poured into the cavity of the endoplasmic reticulum, rough endoplasmic reticulum. Importantly, these are tagged with mannose 6 phosphate within the Golgi apparatus to target them to the lysosome. So, these proteins are tagged with mannose 6 phosphate, indicates that these proteins are of lysosomal. So within the Golgi apparatus, this is being done. As a result, vesicles containing these enzymes bud off from the Golgi apparatus. I told you, Golgi apparatus has two phases, cis phase and trans phase. From the trans phase, these lysosomes which are containing hydrolytic enzymes bud off from the trans surface of the Golgi apparatus. These vesicles now in the cytoplasm then binds with the late endosome. Endosome is formed by the plasma membrane of cell, which is another acidic membrane-bound cell organelle. This is also another acidic membrane-bound organelle. The late endosome has proton pumps within its membrane that keeps its internal environment acidic. The low pH causes dissociation of the protein from the mannose 6 phosphate receptor. This receptor can then be recycled back to the Golgi apparatus. The phosphate group is also removed from the mannose 6 phosphate tag to prevent the whole protein returning to the Golgi apparatus. The late endosome can eventually mature into a lysosome. The late endosome mature into lysosome, functional lysosome. After it has received the enzymes from the Golgi apparatus, it is formed. Then function of the lysosomes. The hydrolytic enzymes contained within the lysosome allow foreign particles to be destroyed. So if a cell receives any foreign particle or foreign body, that would be mixed with hydrolytic enzymes of the lysosome. Thus, the foreign particle is destroyed. Lysosomes play an important role in phagocytosis. What is phagocytosis? Cell eating. Phagocytosis is a process by which cells eat the foreign bodies. So by the process of phagocytosis, cells kill foreign bodies with the help of lysosomes. When macrophages phagocytose foreign particles, they contain them within a phagosome. The phagosome will then bind with the lysosome to form phagolysosome. Phagosome is a body, membrane-bound body within which the foreign body is present. This phagosome is mixed with lysosome, forming phagolysosome. Within the phagolysosome, the foreign body is digested. These enzymes are critical in oxygen independent killing mechanisms. Lysosomes also help to defend against pathogen entry via endocytosis by degrading pathogens before they reach the cytoplasm. So any foreign body which enters the cell body is killed with the help of lysosome before it causes any damage to the cell. So this would be like this. This is lysosome. So this is phagosome. Phagosome is a body which contains bacteria, foreign body. Any foreign body containing the membrane-bound structure is called phagosome. So phagosome is mixed with lysosome. Can you see here? So lysosome containing hydrolytic enzymes. These hydrolytic enzymes are released, and these enzymes acted upon the bacterium, foreign body. Now it is digested and the residue is excreted out. Debris are released out through exocytosis. This is endocytosis, exocytosis. This is how the lysosome play very important role in not only digesting the substances, it also helps in defending cell body from foreign organisms, from pathogens. Pathogens are killed by this process. So lysosomes are very, very important for killing foreign body. So this is about the lysosome. So lysosome, Golgi complex, the endoplasmic reticulum, vacuoles, all these together constitute endomembrane system. Their function is interrelated. The endoplasmic reticulum produces the protein or lipids. Protein is synthesized by the rough endoplasmic reticulum that would be sent to Golgi apparatus. From the Golgi apparatus, the lysosomes are formed. So all are interrelated. So the assignment on this chapter is explain the structure of lysosomes. Explain the structure of lysosome. So in brief, try to give its structure. So lysosome is a membrane-bound vesicle-like structure synthesized by the Golgi complex and containing hydrolytic enzymes which are necessary for the digestion of food substances as well as killing foreign bodies. So explain the structure of lysosomes. Thank you.